I'm speaking today with Dr. John Moult. Dr. Moult is renowned for his work in computational biology, including protein structure prediction. He earned a bachelor's degree in physics from the University of London and a PhD in molecular biophysics from the University of Oxford. He is currently a professor at the University of Maryland's Institute for Bioscience and Biotechnology Research. Dr. Moult co-founded CASP, the Critical Assessment of Protein Structure Prediction, in 1994. Dr. Moult, thank you so much for joining me today. I'm so excited to hear from you about your field and your career.
Oh, a pleasure.
So I was hoping, Dr. Moult, that we could jump back in time a little bit. I think recently there's been so much attention to CASP and the 2024 Nobel Prize winners, but I'm curious to hear about the early days of protein structure prediction. So I'm curious to hear you share, how did you first become interested in computational biology and in specific predicting protein structures?
Well, in terms of computational biology, it turns out I'm not a very good experimental biologist, and so I gradually drifted into it. But interest in the protein folding problem, when I was a graduate student in the 1960s, there was work by a man called Christian Amundsen, who subsequently got a Nobel Prize, showing that for proteins, you didn't need any information except the sequence of the amino acids that make up the backbone of the protein to get the three-dimensional structure. He did this experimentally. He demonstrated you could take a protein apart, as it were, and with no biological machinery, put it together. And so that immediately crisply poses a problem. If all I need is the information of the sequence, surely I should be able to calculate what the three-dimensional thing looks like. And I didn't have any time as a PhD student, but at various points after that, I became more and more involved in, well, how could you do that?
Okay. I'm wondering, as you're talking about that, were there scientists who had come before you in the field of protein structure prediction who were especially inspirational to you?
Well, this was really the start. I think it's a long history, but understanding what proteins are like, that you could really do this, took centuries. And we'd only just in the 60s got to this, it should be possible to do this calculation. So although I wasn't very active at that point, there weren't other people sort of role models to build on. We all sort of grew the field together after that.
Okay. All right. Well, thank you. So earlier I mentioned the Nobel Prize that was awarded in chemistry in 2024. This was awarded to 3 scientists in your field. And I'm hoping that you can share a little bit for our audience about, why is it so important that we understand gene expression and the dynamics of protein folding in modern biology, medicine, and other fields?
Well, I think those two things are at the heart of how biology works. When you turn on genes, when you turn them off, how that affects the behavior of particularly cells, but then, of course, organisms in general. And, of course, many genes code for protein molecules, and protein molecules are very much the machines of biology. I guess we'll get to this, but they modify, recognize, transport, all of that stuff. Not quite all, but nearly all of it is done by proteins. So you can't do biology without understanding how those things work. I'm not so sure that what we'd be good at is understanding the dynamics of protein folding. We're more focused on what does the folded molecule look like, and so because of what it looks like, what does it do?
Okay. So if we go back in time, like in the early part of the career, I think you've started getting at this, but what were some of the biggest challenges in predicting protein structures in the early days?
So I was a physicist by training, and I thought, as most of us did, of it as a physics problem. You have thousands of atoms. Each type of atom likes to interact with other types of atom and doesn't like some of the other sorts it could get together with. And so you have to be able to describe very precisely the energy of interaction between different types of atoms. And that's still an ongoing field of development, so-called force fields from which here are the atoms at a particular position. How do I calculate what the energy of that is? And then you want to find the lowest energy state. That's the physics of the thing. And if you think about the number of conformations even a small polypeptide chain can adopt, it's super astronomical. Something, numbers a bit rough, but 10 to the 50 different arrangements for even a fairly small protein. So how can you sort through 10 to the 50 possibilities and for each one get the energy? And so you've got this map of energies, and this is the one you think the protein. So these are both, in different ways, very, very challenging problems.
So I'm breaking this down, like it's just incredibly complex. We have these different functional groups. We have atoms that may be attracted to other atoms. There may be other possibilities where there are repulsive forces. And just putting it all together for these complex molecules is a big task.
A big task, and actually, just clearly you can't do the folding problem, getting all these different conformations by brute force. It's just too many. And so you must have ways of developing shortcuts of some sort to get you there. So it's not brute force. You need some kind of algorithm or idea about how real, was something we were obsessed with in the early days, how do real proteins solve this problem? Most proteins fold, varies, but in the order of a second. And you can try one conformation about every 10 to the minus 12, 10 to the minus 13 seconds. If you put those numbers together, it would take longer than the history of the universe to fold the first protein. So in some sense, nature has found a way of solving the search part of this problem, right? And so a lot of attention to how does that happen? How could you develop computer algorithms that would do the same thing?
Right. I never thought about the timing aspect of how quickly the proteins fold. That's interesting. So those early iterations of CASP, how did they improve our understanding of proteins? So I'm thinking, the pre-AI revolution that we'll get into a little bit later. Like, what were some of the early innovations that kind of moved the field forward and allowed it to eventually get where we are now?
Yeah, I guess, I'm not sure we understood proteins so much as understood how well the ideas we were having for calculating the structures, bearing in mind the sort of things I just said, how well those ideas did or did not work. So CASP tries to get real clarity. So I got an idea, I test it. Can I test it as a community? Can I test it very rigorously? And I find out it was a really lousy idea. This is extremely useful because I, in particular, can get stuck on bad ideas for decades, and many of us are like that, right? So being able to say, what a lousy idea, but what does that tell us about what might be a better idea? What might be a better algorithm for trying to tackle this problem? And so I think at the beginning, we really moved quite quickly in terms of that kind of evolution. Some areas really didn't get very far. It was sort of more of an academic pursuit moving forwards in terms of a practical solution. But in other areas, we did advance to useful approximations for some types of protein structure. And so it was that progression, from my point of view, that was very satisfying, that we try to work as a community and as rigorously as possible, we can actually move things forward.
It must have been at times like hard to find that motivation to keep going. It feels like it must have felt like such a big challenge. And there's part of the motivation maybe to find the way forward, but it had to be at times challenging to keep going and not say, well, maybe I should refocus my career and do something else.
Yeah. Well, I think there are several aspects to that. First of all, if you're going to do science, what's the most important thing you could imagine doing it? And then, your ego and your optimism and so on keep you going when you probably should have given up. But along the way, there are sort of small satisfactions, right? And so even the small satisfactions keep me anyway, motivated. Yeah. So there's a few things, the over-optimism, over-self-confidence, and then the satisfactions on the way. I think that applies to many scientists, right? It's not.
I'm sure that it does. I think that, if I think about the way that society may view science. I don't know that we always have people getting that behind the scenes look. We hear about the big innovations or the big discoveries without maybe knowing all the backstory and all the years of trial and error and that process.
Right. I mean, one of the nice things for me, having been in this community a long time, and again working very closely with other people, more closely than science usually does with other people, is you get this kind of sense of the sort of dynamics of the thing and how ideas ebb and flow and sort of enthusiasms come and go. And it's a really extraordinary process. Because most science is like it's not somebody in a back room. It's a community of people working towards a common goal, sometimes a bit antagonistically, but they're all trying to go in the same direction.
Well, I mean, science is done by people, right? So we have all kinds that are going to be involved in the scientific endeavor. Okay, so maybe we transition now to talk a bit about the revolution, really, that artificial intelligence has brought into your field. I'm thinking about AlphaFold, RosettaFold, as some of the big names in the CASP competitions. And how did these change the playing field, or how did they change the dynamic of what was happening in protein structure prediction?
Well, different ways of looking at it. And one way of looking at it is that those methods made use of all the experimental data that have been accumulating over the years while we were playing about with our physics models. And so there's a thing called the Protein Data Bank, which has all the experimental structures in, very nicely organized. There are very nice sequence databases. And so when you've got enough of this data, you can think about using neural network methods that need a lot of information to train on because you've got the information there. If you look at what happened with AI methods here, for those of us coming from physics, it's a bit disappointing because you've accumulated all this data and it's become irrelevant. All of your understanding has become irrelevant. Actually, I don't see it that way, and this is controversial how you should think about these methods. Going back to the two problems I talked about, the search problem and the accurate description of the energy, you can see these networks with billions of connections as allowing you to have a very subtle energy description, much more subtle, much more nuanced, much more representative of the true physics than the 200 parameters or so that we typically used in the old physics days. And it's known that these networks essentially transform things into a simpler space. And so the search space now becomes, it's oversimplifying, but a sort of bowl you can just go down the energy surface once you've got this representation. So the way I like to think about them is the sort of physics things we tried to do were much too crude. And these sorts of networks allow us to solve the problem, but we're still solving a physics problem. And we've used all of that data to learn a much better representation of the energy. I think, as you may be aware, there's still a lot of uncertainty about how you should think about what these large neural networks do. And it very much depends on the application. But we've tried experimentally, in a computational sense of experiment, to probe some of those things. And as far as I can see, that really is what's happening, this very nuanced description. It's not that, some people try to present it as you've remembered all the known structures and you just sort of look one up. First of all, you don't know all the structures to remember. And secondly, the network doesn't seem to behave like that. It seems to behave as if it's learned the principles.
Okay. So it's more flexible than if I had a particular, I want to reference something here. So if I had a particular, this is actually from PDB. They have their educational resources. And this is a model of insulin. So if I had a specific sequence of amino acids that's producing an alpha helix, you're saying it's a little bit more complex than just saying, well, whenever we find that sequence in a different protein, it's always going to produce that same alpha helix. Like there might be a bit more to it.
Well, 2 points to that. If you don't often find, the helix is, let's say, 20 amino acids long, you will not find normally a repeat of 20 amino acids in a different protein, just so unlikely. There are a few cases like that where you have, 5, 6 amino acid repeats. And so you can look across known structures and say, is that 5 or 6 always the same? And the answer is no, it's not. The environment will often change what that little chunk does.
Sure.
And so you've got to be looking at the full environment to see what's going to happen, sort of thing.
And if I'm understanding correctly, you're saying that these systems are actually, they're able to do that. They're intelligent enough that they're doing that to some extent, we think?
Yes. I mean, they're able to essentially sense all the interactions around one amino acid, and they do think in terms of interactions. That's how it all got started, representing what amino acids are likely to be next to each other. So it's not so much that you can get a 3D output, but internally they're thinking in terms of relationships between neighboring amino acids, neighboring atoms, and what energies they represent. And again, you can do that very extensively and very subtly if you've got a big enough network.
Right. So is, I'm thinking a little bit about like limitations and existing challenges. So it sounds, if you said if you have a big enough network, like computing power, right? Is that just still somewhat limiting what we might do?
ChatGPT computing power, this is peanuts. I mean, it is significant, but there are two parts to that. One is how much computing power do you need to learn from the original data to get the network. And that's, you need good computer resources, the sort of thing a company can afford much more than an academic, but it's not sort of enormous. And then once you've developed the network and you've installed it on your computer, the compute for a new protein or whatever is almost trivial. It depends. You could spend more. Sometimes it's worth spending more, but it's not really the limitations in this application of AI are not really the amount of power you can bring to bear.
Okay. So I was wondering, I think next we can talk a little bit about, or I'm curious to talk about the connection to the central dogma and protein structure, mutations. So in class, my students learn about the classic pathway of gene expression. So we start with the gene, we have DNA, which is used to produce an RNA molecule, which is then transported to the ribosomes where proteins are built. So I was wondering if you might say a little bit more about the role of protein folding in determining the function of the protein.
Right. So to me, this is one of the most miraculous things in biology. So as you said, you've taken the DNA, you've turned it via RNA into this string of amino acids on this flexible polymer, and you've got usually, it's oversimplification, that you've got there's this one folded conformation where you've chosen the sequence of amino acids to stabilize that conformation. And what that confers mostly in terms of biological function is recognition of other molecules. And so if you think about sort of the proteins most people are familiar with, so proteases in your stomach, for example, trypsin and chymotrypsin and so on, they start out as these wobbly peptides. You fold into the folded shape and you create some complementary surface shape to another peptide. In this case, that's what you're going to act on. And you position the residues that are going to do the chemistry in the right places relative to where that binds. So all of those things are determined by the three-dimensional folding that allows you to sort of move these atoms around in the business end in this very finely tuned way to recognize across biology almost anything and to do and limitations, but a huge number of possible chemistries with enzymes, for example, or to recognize things and then have a mechanism by which you transport them and dump them somewhere else and so on. So it's the purpose of the three-dimensional structure, if you like, is usually to confer recognition by making a specific and activity. And so you've transformed from this dumb linear thing to this beautiful three-dimensional thing with this sort of, active property.
Right. So we're talking about the active site of an enzyme where it can interact with the substrate molecule, or a specific part of a protein that can serve as where a ligand interaction can happen and trigger cell communication process, signal transduction or whatever processes happening inside of a cell. Okay. Next, I wanted to ask you if you have some favorite examples. Maybe we could do one of a disease that's caused by a mutation that would alter the protein folding process, and how you would describe the relationship of the gene, the relationship, of the mutation to the gene expression and protein folding and the effects of the disease.
So, if you're thinking about disease, the large majority of mutations act by destabilizing the folded structure. So you might have, the hydrophobic, the inside of a protein, mostly nonpolar things. You introduce a charged lysine or something right in the middle, and you blow the thing apart energetically. And so you've lost the three-dimensional structure. So by definition, you've lost the properties to bind to things.
Sure. So then it's almost totally non-functional at
That's
that point.
You've lost function because you've lost structure, putting it simply, sort of thing. There are cases where there are diseases cause a conformational change, or at least control a conformational change. So if you look at quite a few mutations that contribute to cancer, they affect the function of kinases. So kinases involved in all sorts of processes as a particular class of enzymes, including, for example, cell replication. And you don't want kinases that are involved in cell replication acting all the time. You only want it when you've decided to replicate. And so those are controlled by a process of phosphorylation of some bits of the polypeptide chain. And that drives a conformational change. So this is not the mutation yet. The normal process drives a conformational change that turns the kinase on or off and helps in, let's say, again, replication. So there are mutations which drive that conformational change without doing the phosphorylation control step. So they turn the enzymes, they alter the accessibility or the openness of the binding side of the enzyme a little bit, as you should do just with phosphorylation by introducing a mutation. So now it's permanently on, and the cancer says, goody, goody, I can keep churning out new cells at a much higher rate. Right. So the kinases and the regulation of that, I think, it's a large class, but that's a very nice example to look at as a conformational change.
Okay. All right. So can we talk a little bit about discovery or formulation of ideas for new molecules? I think there's been a lot that I've been hearing lately through news, through podcasts, about using AI models like what David Baker and his team have produced to contribute to developing new molecules that might be used for discovering new drugs. What are some of the exciting developments that are happening with this type of event in the field?
Right. So, of course, there's this forward problem where you have a sequence and it's a natural one. You want to know what fold it makes. But now I might want to say, I'm going to inhibit the action of some protein involved in an infection, let's say. And so can I do the reverse? Can I say, again, it's this recognition thing. Can you see my hand okay?
Mm-hmm. Yes.
So here's a protein I want to stop doing its job. It's part of an infection process again, perhaps. Can I design, and I've got a surface here, can I design a protein with a complementary surface to that one? So what I start of my design is I know what surface I want to create. And now you can run the sort of AI algorithms we were talking about backwards to say what sequence would give you that folded three-dimensional structure.
Okay.
So it's the sort of the reverse of the folding problem, the design problem. In practice, you need slightly different approaches, but that's almost what you're doing. And so AI methods have been developed very much related to the folding ones to do that reverse process. As you mentioned, David Baker has been really the leader of this for a long time. What was the rest of the question?
I'm just wondering, so that process, so you're creating a molecule to interact with another molecule, which, so this other molecule might be one that's having some sort of negative impact in the body. Right. So we're doing something to bind to it.
Mhm.
how many molecules are they, how many potential candidates are they generating that are
So it's gone
candidates that
down
might
a
fit?
lot. As the methods have got better, you need to do fewer and fewer experimental trials. It used to
Mhm.
be, well, these methods kind of work, but they're only going to work, I don't know, 1 in 100, 1 in 1,000, and so you have to do a lot of experimental trials. These
Okay.
days, the numbers, it varies with the problem a lot, actually, but
Mhm.
1 in 10, if you're lucky, would be enough to actually find something that works. If it's a simple, again, very probably dependent, but there was recent publicity about snake venom inhibitors.
Yes, I
But
saw
it's
this.
simple. You've got a fairly simple protein to design just a binder to, and I don't know the details there, but I would guess
Mhm.
the number of experimental trials. Once you so one of the nice things in design is you can do this process, go backwards from what you want to bind to, get the amino acid sequence.
Right.
Then you can go back to something like good old AlphaFold and test whether you actually get the right structure out from that sequence. You used to have to do the
Okay.
experiment, right? What does this actually
Mhm.
do? Now you can run AlphaFold and see if it looks like it does the right thing. And if it does, it'll probably work experimentally. So that's made a huge difference to the number of trials you need to do.
Okay. Wow. I mean, so it's fascinating that you have this one AI system creating something that you're going to feed into another AI system to test to see if it's
Yes,
going to
yes, yes, yes.
hopefully do what you think it's going to do. That's kind of, yeah, that's fascinating. I need to move a little bit here. I'm getting all kinds of glare in my.
Right,
I'm going to
thanks.
just close my window for a second. Okay. Hopefully that'll be a little bit better. I was kind of getting blasted with the sun,
Mhm.
which is, as a teacher, I have been in a lot of classrooms without windows. So I'm thankful for my windows. It's hard. It's like you're in a cave. I don't know what the lab settings are like that.
It depends. Yeah.
Yeah. So here, okay, we were talking about the snake venom, developing these molecules. So at the point when there's a good candidate molecule, it's been generated, it's been fed into AlphaFold, it looks like it's going to do what you want it to do. What's the process like then to actually make it in real life and put it in cells to test to see if it's going to have biological
Um,
activity?
Pretty established technology. So you make the corresponding piece of DNA to the sequence you want for the amino acids, you clone it into a cell line, and you get the cell to churn out the protein and then purify it from all the gunk. If you're lucky, you can set up a system where you'll export your protein so it's sort of fairly clean in the surroundings, and then
Okay,
sort of standard biochemistry stuff, running it over columns and so on to
sure.
take out things you don't want.
So is that typically we're using bacterial cells, like producing insulin, but we're producing these new proteins?
Sort of
And
idea.
then,
Yeah, the sort of workhorses for this, and so, but it does depend on the protein a bit. But
mhm,
one of the things about these design proteins is they tend to be more robust and well behaved than natural proteins, and so
really?
so they're sort of easier to work with, and you get away with fairly simple expression systems. You don't need some fancy eukaryotic cells with all sorts of refoldases and what have you. Depends
Okay.
again, but there's a general rule.
Do a lot of the designed proteins, I didn't have this question in, it just popped into my thoughts as you were talking about this. Are a lot of them including multiple different polypeptide chains and they have quaternary level structure, or is a lot of the design?
At the academic level, yes. So if you look at David Baker's work, he's sort of making materials
Mhm.
almost. First of all, started making sort of bigger and bigger oligomers, and now has sort of things that fit together. And there's obviously a major kind of application there in building new materials with
Mhm.
proteins. But typically, again, if you go back to things like the venom example, you can get away with it where we've got to anyway. You can get away with it with a single protein. You can
Okay.
imagine applications where you might want some sort of carrier protein that took your business protein into the cell and then dropped it like a lunar
Mhm.
lander or something. But that sort of technology is not so advanced as these relatively simple things.
Okay. So many of them are single polypeptide chains folded into a tertiary structure.
That's right.
Oh, okay. All right. I think we're getting close to the end. I was hoping to talk about the future a little bit. I remember back in the day, I took an educational course on futurism, and kind of thinking about where things might go in the future, 20 years, 30 years.
Well, you're right.
I wish I could find my old notes and see what things my professor had talked about. But I'm so curious to hear from you. Where do you think this field is going in the next 5, 10, even 20 years out?
Yes, well, this is a dangerous question, obviously, but nobody will remember this video. Well, maybe they are. So I think, if you think about the sort of things we're talking about, computational models in biology, I think that they are going to be the next big thing in some sense. I think if you look at biology, it's been an experimental science with some sort of computational tools as a backup useful. AlphaFold is perhaps the first example of this, where now you can just think about proteins as virtual objects. You often don't need to make them to work out their properties, their folds, and so on. And you can think of extending that to, first of all, cells. And so there's a very serious project with a lot of investment to make a virtual cell. So the idea is you have all the components of a cell. You actually use AI methods to calculate how these all interact with each other. And so there's a lot of data being generated to support that sort of thing. And then if you can get this to work for something simple, you can say, well, what happens in disease? This is how the cell goes wrong virtually. And then virtually say, well, what if I poke this? Would this affect the disease phenotype in some way I'd like? And so rather than doing experiments in the lab on cells and so on, you'd have this kind of representation. And of course, that's very much a trend in sort of medicine as well, so-called digital twins. And one can imagine each of us having a digital twin. And when a physician is deciding what drug to give us, he doesn't give us the wrong drug first. He tries it on our digital twin and says, aha, that's a bad idea. What about this other one? And so,
oh no,
kind of thing.
The digital twin. We better not do that.
The whole thing becomes like a video game, right? That you're interacting with this virtual world, which has properties of the real world.
Oh,
and so, again, I think the sort of AlphaFold thing is just a tiny start of that. But you could just outline there are many areas where people are trying to do that, and at some level, it's going to be powerful. I mean, is there really going to be a virtual version of an individual? You go to and it's you, and I don't. That's pretty futuristic, but it's not inconceivable at this point that one could get there in another 2 or 3 decades.
I'm going to be thinking about that and processing that for a while, I think. And it starts to get into the field of ethics. And do you see that there are other ethical considerations you feel like are really important moving forward in this field?
Well, I think there's 2 kinds of ethical considerations. One, it's been much talked about, there is the possibility of producing bioweapons with the sort of technology we've got and we'll have more of. And so how do you, as a society of humans, regulate appropriately to try and control that risk and try to get appropriate international agreement? Like with nuclear weapons, we've, for 70 years or so, we've managed to not blow each other up because we realize it's a really dangerous thing. Will we be able to do that kind of realization of the dangers with this sort of technology? And then, of course, the other the sort of more familiar ethical issue is you've got all this power to change biology. What's acceptable in that regard? Should, for example, parents be allowed to make, and this is already happening, 50 embryos and have their genomes screened for greater intelligence or greater athletic prowess or whatever the parents happen to fancy? What are the limits in that sort of direction versus the limits of have the embryos screened and only use the ones that don't have some disease tendencies? There's a whole spectrum of choices like that that I think are a major ethical. Actually, I teach human genetics, and yesterday we had the class talking about this, so sort of fresh in my mind. But I think that's one of the main challenges from an ethical standpoint.
It makes me think about, I don't know if you've seen the film Gattaca that came, I think it's over 20 years old now, but they were considering a future where parents could, select traits of their children, and, brought up a lot of those ethical considerations. And I remember watching it back in the mid to late 90s when it came out, thinking, well, this is so far away in the future. This is so unrealistic. And now it's maybe not, at least technology-wise, maybe not that far off of what we could start to think about doing. Yeah. Okay. I'm sorry. I lost myself in a thought there for a moment. As a teacher of high school students, one thing that I really love to ask researchers like yourself is, what would you say to a student who's in high school, or maybe is an undergrad, their first year, second year student, and they are really interested in this field? They would like to pursue it. What are some of the top things that you would recommend for them to do, or to consider to try to set themselves up for success in their academic career?
Yes. Well, I think it helps if as early as you can to know what you want to do. I mean, I certainly didn't know what I wanted to do, so it's very impractical advice, actually. But if you were thinking about a career in research biology, I think that I'd say two things. I do believe in this virtual biology we just touched on, and the other is synthetic biology. So we mentioned the work of David Baker starting to make materials and so on. I think my imagination fails, but now we have control of what proteins are like, what they can do, and so on. One will be able to do all sorts of new machines, which I can't get the limits of this, right? But which will replace the sort of machines we're familiar with. So there might be machines which are designed to go around in your blood and monitor what's going on and spot early signs of cancer because they're really in there. Totally hypothetical, right? But in general, the idea of synthetic biology, I think, is, if you're interested in applications, that's a really interesting thing to do. If you're interested in understanding biology, I mean, I would go for the virtual world stuff, because to me, biology is, I could just, this bit interacts with this bit and it's all fine. It just is mind-boggling how complex these systems are. And I think we don't have the theoretical framework actually to comprehend them as systems. And I think the virtual stuff will start to allow us to do that. And so if you're interested in, I really want to understand this stuff, the virtual biology route would be the thing I'd be attracted to.
Yeah, I mean, I think there's so many, like, it would be, I think, very exciting to be a young person now who's just on the cusp. Like, so many amazing things.
To go into biological research, for me, has just been an enormous privilege to spend your career doing this great stuff. And I assume that's going to go on. I assume it's going to get, and this is a revolutionary age for biology, obviously, and we're not anywhere near the end of it. We're just at the beginning of it. So, if I were starting again, I'd, in some sense, do the same thing as a research biologist.
Yeah, it feels like almost like a video game where you're unlocking the next level, and who
Yeah. Good analogy, yes.
Well, Dr. Moult, it has been such a pleasure to meet with you and to learn more about your field. I like to close all of my interviews with scientists with a fun question. Before we do that, is there anything you were hoping to share that you didn't share yet?
I don't think so. Your questions are good, fun questions.
All right. Okay. So I have a fun question now for you. So I like to ask every scientist I talk to this one. So if you could choose to be an animal, any animal for 24 hours, which animal would you pick, and why would that be your choice?
Well, I think there's too many choices, really. I mean, I have a fairly rural setting here. I can look outside and see the animals, so squirrels and foxes and deer and so on. I really wouldn't like to be a deer. I mean, they look bored out of their minds most of the time. But squirrels seem to have a really great time. They're always having fun. They're always chasing each other, and I don't know what they're up to. So I think that would be nice, but I'm not sure it's very sort of profound. It's kind of a frivolous life, right? And foxes look very intense.
Yeah.
But I'm not sure that that's a sort of genuine intellectual intensity, which I would like. So actually, I'm not sure. Do birds count as animals? Because I want to be a bird. I think that the idea that you can be in three-dimensional space and looking at things and moving in three-dimensional space would be a level of freedom that would be really, really attractive. So that's my choice.
Yeah, no, I think that's a really good one. I mean, the ability to fly and, to look down on everything under your own power. We can do this, of course, if we're flying commercially or but the ability to do that just under your own power would be really neat. Yeah, I think a lot of times I've said I would like to be a whale.
A whale?
Yeah, for 24 hours and dive to the depths of the ocean as far down as I might be able to go and to explore something that, I'm probably not going to see as a human. I think that would be pretty cool.
It's sort of the reverse from going up to going down, right?
Yeah.
And, I used to scuba dive, and so there is some of that sense of freedom when you're moving in the water. So, yeah, that's a good one.